So, good afternoon, everyone. First of all, I would like to acknowledge the presence of Speaker of the Day, Ms. Riddhi Salotra. I also want to acknowledge our worthy ma'am principal in her absentia for always encouraging us and to do such activities. Next, I would like to welcome our head PG Department of Chemistry, Dr. Manju Sahni. She has always motivated us to touch new heights of success. Kanya Mahavidyalaya, the heritage and autonomous institution conferred with star status by UGC MHRD Government of India, with top national and state rankings by India Today's Survey of Best Colleges 2020 and Outlook magazine, is a pioneering institution for women in Northern India. PG Department of Chemistry was established in 1950, is a dynamic center for research at the frontiers of chemical science. Our ethos at PG Department of Chemistry is to enlighten the students with scientific temper, practical skills, and confidence for their holistic development. The department's strong record of achievements in chemical research is solidly based on its pioneering advances in chemical research, its success in incorporating these advances into teaching and research programs, and its close relationship to industry. The department offers core courses in chemistry for undergraduate and postgraduate students and has been recognized by DST and FIST programs. The department flourished with highly qualified, dedicated, and hard-working faculty toiling to achieve excellent and sustained records of academics and placements. So it is a regular feature of the department to organize such activities, and in continuation to this, we are organizing KMB International Series Chemistry Chapter, live interaction with Speaker of the Day, Ms. Riddhi Salotra, on the topic Developing Research Ethics for the Better Future. So we welcome you, Ms. Riddhi Salotra. Let me read her biodata. Ms. Riddhi Salotra is a Marie Curie early stage researcher at Queen's University, Belfast, UK. She got involved with an equitable opportunity as a staff member under Marie Curie grant at Queen's University. She got this Marie Curie award, which is a very prestigious salary award that covers all her expenses and provides very good amount of salary. This is a very honorable title for a researcher. She did her master's in chemistry from Punjab Agricultural University, Ludhiana. She has publications in national and international journals. She also got best poster and oral presentation awards in many national and international conferences. Best poster presentation entitled Removal of Chromium-6 from Ternary Effluent Using Low-Cost Leaf Powder in National Seminar on RACS organized by Dunningcliffe Chemical Society, and best poster presentation in international conference on recent advances for quality enhancement of science and technology organized by Faculty of Sciences in collaboration with International Association of Research Scholars Australia. So we are happy and very proud that Ms. Riddhi Salotra is among us, and she will be having a live interaction with our students, and she will also will provide us a virtual tour to Queen's University, Belfast, UK. So over to Ms. Salotra.
Thank you so much, ma'am, for the kind introduction. At first, I would like to thank Principal of Kanya Mahavidyalaya, Professor Dr. Atima Sharma, for providing me this opportunity to deliver this session today. And my special thanks to Dr. Narendrajit Kaur for motivating me to deliver today's talk. So now, let me share screen. So our today's topic of talk is developing research ethics for the better future. Moving on to the introduction, I would like to introduce all of you with the basic sciences research focus. So I assume all of you might be from the basic sciences stream, maybe chemistry, biology, or physics background. So basically, basic sciences at present plays a very significant role in solving all the global challenges of the present century. Here, I have highlighted some of the important goals which around which the research of the basic sciences basically revolves. So all these goals are the sustainable development goals. So basically, whatever the research is going on throughout the globe, all that research focus or it goes to the main point like the sustainability. So the very first highlighted one is the good health and well-being that comes under the biology sector or the chemistry sector. The next one you can see is clean water and sanitation. Again, it covers the chemical engineering or chemistry background. Again, the affordability and clean energy. Again, it comes the sustainability sector or environmental sciences. Then sustainable cities and communities. This sector of research work basically covers your architecture of designing the buildings in such a way that you make use of more use of the renewable sources of energy like solar cells and all that stuff. And then it comes to climatic action, which is the main prime focus of most of the scientists and the researchers around the globe, because you have seen there is a lots of global warming, and due to the emission of greenhouse gases, climate is changing a lot, which leads to the focus of the scientists towards the climatic action, and it actually covers most of the fields. It is an interdisciplinary sector. Then you comes the life below water. So life below water is the most affected part, like the aquatic life habitat by the climatic actions and the deforestation and the pollution. So all these factors, they affect the life below water. So this is also have become a prime focus. And then life on land, again, it covers the pollution part, like the land pollution, the disposal of waste, all this stuff. So the purpose of showing you this slide was just to make you aware about the basic sciences research work, which is being carried out throughout the globe. And you guys are very lucky enough that you are pursuing your undergrad degree in this basic foundation, which actually makes your base very strong to establish a fulfilling and a brilliant research career in future. So now moving on to the basic streams of the basic sciences: chemistry, physics, biology. I have tried my best to highlight the main fields that comes under each of the sector, which covers the important research areas around the globe. So in chemistry, you might be aware with organic chemistry, inorganic chemistry, physical chemistry, analytical chemistry. Now, the new term here is computational chemistry. Computational chemistry basically relates to the software programs. Here, we don't do work in the wet labs. Instead, we do the same experiments, same reactions on the softwares. Then comes the sustainable chemistry. This is a very, very broad field, which covers like different fields. Like if you are focusing on any sustainable project, like it be water pollution, you want to eradicate water pollution, right? So in this term, what will you do? You will cover some biological scientists, some microbiologists in order to remove the contaminants, and then there will be chemists, then there will be technology used. So there will be engineers as well. So it is a kind of very broad field that comes under chemistry. And then photochemistry, that covers the use of light as a catalyst in the breakdown of various compounds. So moving on to the physics part. In physics, there are different fields like mechanics. In mechanics, you can go for the mechanical engineering side as well, or classical mechanics. Then there is astrophysics. Astrophysics is the study of stars. So you can go to the astronomical engineering side or to the simple stars and how the satellites work and all that stuff. Then comes thermodynamics. Again, thermodynamics have two parts: classical thermodynamics and statistical thermodynamics. You might be reading these courses in your semester. So next is optical, and then nuclear and atomic physics covers the particle physics. So there new particles are being developing by the different scientists. You might have heard about the headquarters in Geneva, Switzerland, where the basic foundation of the particle research is moving. And then geophysics, that covers the geography, like devising different techniques for studying the geographical satellites and geographical things around the globe. And then biological section. In biological section, research is being carried out in botany, zoology, microbiology, genetics like biotechnology, then bioinformatics. So here bioinformatics is quite related to the computational chemistry. It is computational biology in simple terms. And here also, instead of testing on it is a very efficient field, and it is very new technology, because it actually don't let you do the testing of the medicines on the animals. You can do it directly on the softwares, like just to see that which medicine is good for which disease. So it is a very efficient technology, and it is very new. And then ecology, that is environmental sciences, and then pathology related to the study of plants. So these are the main three pillars of the basic sciences, and you have like uncountable number of opportunities under these. So now moving on to research. I have been talking about research from the very first slide. So just let you know what is actually research. So research is basically made up of three important phases. Very first is identifying the research problem. Second is developing the solution for that problem. And third is promoting your research, right? So you can see on the left-hand side, I have placed a picture of a flower. So basically, You guys, at the moment, you are at the ground level. You are like, we have planted a seed as an undergrad degree in basic sciences. So you are like that seed who is developing their strong roots by improving your theoretical knowledge related to the different concepts of the basic sciences, right? So after you step out of your undergrad degree, you will be moving into your masters, or maybe the master student will go to the PhD one, right? So you will be flourishing, you will be blossoming like a flower. For that, you should have the proper knowledge how you can pursue your research career in future. So the very first part is, at this stage, this is the very correct stage to identify that in which field you wanted to go to pursue your future career. As I explained you in the previous slides, there are three different pillars. So I hope that all of you guys might have like one of those subjects like chemistry, physics, or biology. So it depends on you. You have to see in which subject you can passionately carry out your future, right? So that will be your developing phase. That will be your prime phase. That is the identification of your passion, in which subject you are more interested. Then you will develop. You will go for your master's or you will go for your PhD. You will develop your knowledge in that particular subject, and then you will blossom like a flower. And that blossoming is actually your promotion. So once you are done with all your basic concepts, you start going for your PhD or for your postdoctoral researcher. Then you start getting your publication, and then you share, you make your network with the other people. So this is actually basic concept of the research. Now, as an undergrad student, how can you improve your vision for research? Like you are just doing your theoretical lectures over here, you have practical classes. So how can you contribute towards your towards increasing your passion for research? So the very first of all, don't go to the lab with the burden of doing practicals. I know you have practical classes at the end, usually at the end. So like you are attending your zero lecture from 8 o'clock in the morning, and then you have practical class around 3:30 in the afternoon. And then you have, oh my God, why I have to do the practical? And even in the practical class, if you are going to the practical class, you will simply sit at the back bench, and then you will start scrolling your Instagram or Facebook, which is not good. So even if in practical class, I have been doing my undergrad here. So during my time, what was the case was like we used to split up into the groups, like group of 5 people. So only the one person in the group used to perform the whole practical, and the rest of 4 people, they will start scrolling Instagram or they will start scrolling Facebook, which is not a good approach. You should have that passion to go in any lab to learn something new in your practical classes. So first of all, be active in your practical classes. Don't take it as a burden. I know as a student, you have that concept in mind, nahi bhai thak gaye hain subah se, kai ko faltu mein khapai karni hai. Your practical class is like your fun class. Enjoy karna hai. But that shouldn't be the case. Okay, try to change your approach towards the practical classes, because practical knowledge is even more important than theoretical knowledge, right? आप लोग घर पे देखते हो. So at this stage, I hope everybody knows how to prepare tea, right? So have you ever read any chapter how to prepare tea, or you have just gone to the kitchen to prepare tea? So unless you go to the kitchen, take your ingredients and start preparing tea, you will get to know how you can prepare tea. That is a very simple example, right? So if you read a chapter on it, how to prepare a tea, I am 100% sure you won't be able to prepare the tea. So the main purpose of telling you this example was, even if you are doing simple test tube experiments with the Bunsen burner, that handling of the test tube with the Bunsen burner in itself is a very big achievement for you guys at the moment, right? So basic lab handling is, you are going to take it from this level. So try to learn as much as you can from your teachers. Even when I was there in my undergrad, I'm here just because of my teacher, my mentor, Dr. Narendrajit Kaur, and one of my mentors back, Dr. Ekta Khosla. She is presently the principal of RR DAV College. So I pursued my research project, which was sponsored by DBT in my undergrad program. And through that project, while I was doing my project, my fellow mates used to tease me, "Hey Riddhi, why are you doing that project? It is just a wastage of time. Like it is of no benefit to you. You're not getting any salary out of it, or you're not getting any exams out of it. So you're just wasting your time. Instead, go to the library, prepare the notes for the semester exams." But no. And just mind my words, like it was a batch of 300 and plus students, and I was the only one person from my batch who was pursuing my research with Dr. Narendrajit Kaur and Dr. Ekta Khosla. And Dr. Narendra ma'am, she has actually trained me with my basic lab handling and with my basic research work. And I'm just sitting in just front of you guys and delivering today's lecture. So just see, this is a very good stage. आप लोग जितना ज़्यादा अपने आप को motivate, passionate रख सकते हो, try to keep in that way. and feel blessed that you are in such a prestigious institution and you are surrounded by such brilliant professors. Try to take as much knowledge as you can from the professors. They are like, they're like those treasure box in which you can take as much as you can. And I really assure you, you will be of benefit of all these basic things that you are learning at this stage. Secondly, engage in the research activities, which is very, very important. Even if you don't get any opportunity in the college, though I know that there are lots of opportunities, but still, if you want to go some out of your college, you can go for the internship programs. In our country, we have different internship programs available for the undergrad students. Go for that. Leave your comfort zone. Don't be like, okay, now I'm already tired with my lectures. I don't want to do anything new. So don't go with that approach. Have an optimistic approach towards your life and towards your future career. So go for the extra research activities. Work on small scale projects. Like you might be having some grants, like your professors would have some research grants. Try to engage in them. So that one step going to the professor, you have to take. You will take one step. Your professors will take 10 more steps in front of you. वो बोलते हैं ना, एक कदम आप बढ़ाओगे 10 कदम आपके प्रोफेसर्स बढ़ाएंगे. I am here just today to teach, just to make you motivate that you have to take that one step towards your teacher. Then your teacher will take 10 steps towards you. And that I can assure you. Okay. And then apart from that, you have to participate. So once you are engaged in all these activities, you will automatically get promoted to the like participating in the conferences and scientific camps. And I'm also like in my introduction, Dr. Narendra ma'am, she has introduced about the conferences and the best post presentation. All those presentations, all those conference achievements I have got during my undergraduate. And I'm really honest in this opinion. I have got all those achievements during my undergrad degree while pursuing my research. So this will establish your confidence. It will grow with your personality. So go for it. Go for the conferences. Even if you don't have any research topic, just go for simple poster presentation. Poster presentation can be of a general topic. It will help you to boost up your confidence and to develop your good network connections in your own field, right? And last point I have enlisted here, like start writing a review article. So now some of you might be wondering, what is this review article now come from? So now some people, they have that passion of doing lab work, learning new lab techniques. But some of the people, they don't have that, oh, this is just, I can't do the lab stuff. But you can, on the contrary, you can start writing a review article, which is like equally very beneficial for you guys. And for writing this review article, it is like a general article. It's like a general compilation of the literature on some particular topic from the previous like 10 to 15 years. And for writing this also, like you will take one step towards your teacher. You will go to your teacher that I want to write this article, and then your teacher will definitely guide you how to write that article, and then it will add up into your publication as well in future. So this is how you guys can improve your vision for pursuing your research career in future. So now moving on to the pathways for better funding. So there are different like paths. How can you achieve better funding? The very first point is better research profile. You should have a very good research background. At least you should have carried out like at least one project in India during, maybe it would be a small scale project of your college level, or it can be some, internship program. So secondly, there is a GRE ETS exam. I don't know, some of you might be aware of this exam. It's an exam, and there are two parts of this exam, general and subject part. So in general exam, you have like two parts, like English and quantitative aptitude. You can go for this exam. It is very good to improve your CV, especially for applying to the universities in US, Canada, and Australia. So you get an extra credit of your this. If you have applied, like if you have written this exam, you get an extra credit in your application while applying for the US universities or Canada or Australian universities. And then publication. As I stated in the previous slide, if you even write a review article at this stage, you will definitely be able to have one publication, and which is a very, very positive point of a student applying to the international fundings. And then IELTS is mandatory. Even if you go anywhere for pursuing your master's or for pursuing your PhD, IELTS is mandatory. And with 6.5 as a minimum criteria, it is just for your knowledge. And then definitely good external links. So how can you establish good external links? Definitely through your conference visits. Okay. Apart from that, you guys are very active on social media nowadays. You have your Instagram account, you have your FB account. So try to make your LinkedIn account as well, and Twitter account, and be active on Google Scholar. On Google Scholar, you will be able to read the articles of the other researchers, what they are doing in their labs. And on Twitter, there are many funding opportunities, as well as on LinkedIn. You can connect yourself with, international professors on LinkedIn, and then they advertise about the vacant PhD positions or vacant master's positions. So it will be a very good benefit to you guys. So now you have got the better funding pathways, right? Now we'll move on to how will you prepare your good CV. So these are the important points that you have to enlist in your CV. Very first will be your academic qualification, like what is from where did you did your bachelor program, and from where did you did your master's program, and all that stuff. And then it will cover your extra research activities that I already stated you, like projects, conferences, in which conferences you participate. Even if you have just participation certificate, you will enlist that as well. Okay. And then your internships, like where did you did your small scale research projects and all that stuff. Then comes your achievements, like if you have got any merit certificate, if you have got any good gold award in your academics, you can enlist that also. Then comes your skills. So now this is the important part, or I would say this is the main backbone of your CV, your skills. These skills will be developed. These skills basically covers your, first of all, laboratory skills that you will learn here at your undergrad level. And then comes technical skills. So now what comes under technical skills? Technical skills are operating some instruments, highly advanced instruments, IR, UV. I'm sure that in your college you have IR and UV instrument. Then there comes like HPLC, NMR, mass spectrometry. All these instruments, even if they are not available at present in your college, these techniques you can cover by going for the internship programs in the like good institutes or in the other institutes out of your college. So you will be able to learn more techniques. You can go that you can get a training for a particular instrument. This will be a very good point in your. This shows that you have a good practical knowledge about your project, about your subject and about the technical parts. And then comes the recommendation. So you have to be, you should have at least 2 recommendations from your professors over here for applying for including it in your CV. So all these are the points that you can include in your curriculum vitae while applying for the international fundings. So now moving on to how will you find the opportunities? So now you have done with the exams, you cleared the GRE exam, you did the IELTS, now you prepared the CV. So now how will you go to the opportunities? So I have here enlisted certain sites. So very first is like for UK, for United Kingdom, there is a site, www.jobs.ac.uk. It is a very, very beneficial site. I must say that you should go for this site. You will just click on this site and you will get like abundance of opportunities throughout the UK with the different filters for different specific fields. And then on Twitter, as I stated earlier, you should make an account on Twitter. So on Twitter, there is a page, Find a PhD newsletter, which is of very benefit. So every day you will get the updates on your Twitter about the vacant PhD position around the globe. So it is of very good benefit to you. And then again, I told you that you have to be active on LinkedIn. So it is good that if you start making connections, so the people advertise their vacant positions on LinkedIn. So it is also a good benefit. Now, in the very first point, I have listed that start listing the universities from state to central, especially for US. So there are 2 reasons for this. The very first reason is in US, the competition is very high. To be very honest, the competition is so high that we as a student, because due to the education system of our country, we focus more on the theoretical concepts than the practical things. So I would be like an honest opinion, I would say that if you want to apply for the US universities, though there are many funding options available there, but I would suggest you that start enlisting them from the state universities and to the central universities. So first of all, apply to the state universities, and then you can see your progress. If you are getting selected, you can go to the central universities. And second reason for like not going from the central universities is that in US, every application charges you like around 6,000 to 7,000 rupees, which is a lot for single application. So my recommendation would be like you go from state universities to the central universities instead of going from central to state. And then from Europe, for European nation. So at first for European nation, the main 2 funding schemes are organized by European Union and UKRI. So under which first is Marie Curie funding, in which I am presently, and second is ERC grant. These are 2 of the prestigious funding grants which are available in the European nations. So you can apply it. It is a very good award. You will have an interview of 3 different filters, and it will be a good benefit to you. And then apart from all these funding opportunities, there are some exchange programs available in our country with the other nations that actually cover your whole tuition fees and expenses. And these programs are organized by the Ministry of Health, Human Resource Development by Government of India. So you can go to their website and you can search for the funding exchange programs. You will find like lots of programs, and you can apply, like even if you want to do your PhD in India, so you can go to one of the good university which are involved in this exchange programs. You can do their PhD in India, like half PhD will be in your India and half PhD or your master will be in some foreign nation. So it will be a good exposure to you in that case as well. So these are all the points through which you can look for the different opportunities. So now moving on to, okay, sorry, I skipped one point over here. Yeah, for European nation in general, apart from this Medicare funding, there is other technique to apply for the funding is there are no charges, but for European nation, you can search through file of the different professors and then you can write an email to them. Okay, so how you can write an email, I will tell you in the following slide. So writing an email is like you have to describe your CV. It is like a short reflection of your CV to that professor, to that unknown person. So your email should be very much strong enough. Like a you person, like if you are writing an email to any professor around the globe, it is not only you. Just keep that thing in your mind that you're not only the person who is sending the email to that person. You are competing with other lakhs of people around the globe just to achieve that person's attention in general. So your email should be strong enough to attract, to draw the attention of that particular professor. So it should in your email start it with your introduction, brief introduction about yourself, any test you have cleared, any achievement, and then start with your research background in which you are interested, and then show your interest in their research group. And for that, you have to read at least one or two articles of that research group, so that it will make your email to look more strong to that person. That okay, this person has read my paper, so you are more interested in that person's research group. So for that, I will share a document with Dr. Narinderjit Kaur. She will share with you guys. I have a an outline of the cover letter. How can you make that email? So it will be a great help to you guys. And then is developing research proposal. So now if you have emailed the professor, you have got selection. The very first stage selection is they will ask you to develop research proposal. So now how will you develop research proposal? So there are two cases here. 20% of the people, 80% of the people, they will already assign you one topic in which you are going to do research. So it is much easier for writing the research proposal for already proposed topic. So even if you have already proposed topic, you can just write the research. You will just write the research proposal. You already have the title. You have to just you have to just read more literature on it. You can propose the goal of the project, and then you can frame research strategy. Just you have to show that how will you carry out that research program in future. So for framing the research strategy, you can go with the background of the research. Okay, just looking for the literature. What is this research about? Then you have to show the relevance, like what you have done in your past in your research. How could it relates to the present research work? Show that relevance. Then you can go for the approach. Approach is basically you have to show them how will you carry out this research work in future if you are given this project. If you have been selected under this funding, how will you carry out this research project? And then comes the resources. In resources part, you can include like what you expect from your professor or what you expect from the funding body, that how much resources they would apply and how would you carry on with those resources in future. So this was only for that 80% cases in which you already have the project title. Now comes that 20% cases in which you don't have any research title. They will just ask you, just write a general research proposal. So what will you do in that case? You remember in the very second or third slide, I showed you that that flower diagram. Exactly. So you have to use that diagram in order to establish a research title. So what will you do? For example, let's suppose. So in that research title, I told you there is an identification of the problem, and then there is developing the solution, and then there is promotion. So now how will you identify? Let's suppose you, like as a chemist, I'm a chemist. So let us suppose in chemistry, I go to the sustainability side and then I just think about that, okay, there is a water pollution going on. So I wanted to remove some organic pollutants from the water, right? So that becomes your broad perspective. Then you start refining it. Okay, how will I remove the organic pollutants from that? So that while researching on that, you will try to make your topic more and more specific, and then you go to that developing phase where you get your title for the project. So you can apply that diagram in actual just to make that research title. So once you're ready with the research proposal title, then you will be able to make the whole proposal in a very efficient manner. And these are the all points that I have enlisted here, which you can add it in your proposal as the headings, and then you can write in the paragraphs in between. For this also, I will share the document with Dr. Narinderjit Kaur. She will share that document with you. It will be a great benefit to you guys. And then moving on to now, you already submitted your research proposal. Let's suppose you got the interview letter, right? They have given you a call for interview. So now the main part is how will you attend a Skype call, because you will be having international fundings. So you can't go to that country to give in-person interview. So definitely it will be a Skype interview, and it is quite different from the in-person interview. At very first, you should be prepared with the questionnaire of the interview. For that questionnaire, you can just simple Google. You can ask for the questionnaire related to the interview. You have to prepare all those questions in general. Secondly, keep this thing in mind that while you are giving an interview, Nobody's testing your knowledge. Nobody's testing your skills. They have already seen all your skills, all your knowledge. That is why you are here in the interview round. You just have to show them your potential to carry out the present research work, okay, and show your confidence that you are able to handle every situation. So the very first point I have written here is that they will ask you to define your background. So first of all, be confident enough while presenting your background. What is your background? What are your academic qualifications? What was your research that you were carrying out in the past? Then comes, you have to, like with your answers, you have to show that curiosity, that passion about your work to the interviewers. Then comes, even if you don't know the answer of any question, don't fumble. Don't say like, if it does like, don't do like that. Just simply say no, sorry, I don't know the answer. That's it. It is a very honorable answer for the interviewers. Okay. And since it will be an international interviewer, so there are chances that you don't understand the accent of the person, because there are different people with a different accent around the globe. So just make sure that stay calm, and you can ask them like very politely, just ask them, please, can you please repeat the question? And they will definitely repeat the question. So don't fumble, right? Just don't pretend yourself that you are very much very much you are afraid of giving the answers, or if you, even if you're not understanding the question, what is a fun of giving wrong answers? So it is better that you ask them that kindly repeat the question. Okay. And the last thing is, always in every interview, the panel will ask you that if you have any question, just ask. This is a very positive point. If you ask a question, let it be a general question, like even if you are going for any research interview or any PhD interview, you can just ask the question like, how many conference visits could I have in a year? Like any general question. Like this, you asking a question to the interviewer panel becomes a very positive point in your positive point of the reflection of your personality. So it's a good thing, right? So this is how you have to prepare for your Skype interview, and this is a procedure for all that. So this was the talk for all of you guys, just to motivate you to pursue your dream PhD with the brilliant fellowships around the globe. And now I would hand over the talk to Dr. Narendrajit Kaur. She'll play the virtual tour of labs at QB for you guys.
Thank you, Riddhi, Miss Riddhi. So I'm showing now the virtual tour. I hope my screen is visible. Just.
First of all, welcome to our iconic Lanyon building. This design can be seen across the city and was designed by Sir Charles Lanyon. Across the road is our brand new Students' Union building. This building will be opening in June 2022 and will have state-of-the-art facilities for our new students coming in. We're so excited that incoming first years will be some of the first to see our brand new building. Across there, we have our Student Guidance Centre. This is a centre in which our students can find all the support and services they need to help them during their time at university. From mental health and wellbeing services to disability services, help with finance, everything's housed in that one building. Over here is the Whitlow Hall. You'll need to remember this building because this is the building where you will be graduating in just a few years' time. This is the Maclay Library that was opened in 2012 by Seamus Heaney. Upstairs on the first floor we have the C.S. Lewis room, which has glass engravings of quotes of C.S. Lewis and has a replica of the wardrobe door from The Lion, the Witch and the Wardrobe. We've just arrived at Queen's PEC, which is the home of Queen's Sport. Come on in. Queen's Sport is one of the leading providers of sports and leisure facilities in Ireland. Queen's Sport has state-of-the-art facilities, including fitness suites, a 25-metre pool, a diving pool, and outdoor training facilities. Thank you so much for joining us on this tour. We're really looking forward to welcoming you at Queen's.
So this was just the outer tour of Queen's University. So now I will show you the virtual tour, which was videographed by Ms. Riddhi Salutra. I hope my screen is visible. Is it visible now?
Yes.
Now I'm taking you to the chemistry labs where we do our research work. So moving on to the first section, this is the Photocatalysis Lab. So here the people, they use the photovoltaic cell to do the breakdown of the organic compounds into smaller components. Moving from here, so these are the office spaces where the students usually sit to do their work. And then moving on to the other labs, this is the nanochemistry lab. Here also the students are working. And then so now we are moving on to the organic chemistry lab. This is synthetic organic chemistry lab. So these are the different hot plates and the different apparatus hanging over here. And here this is a roto-evaporator. Basically this instrument is used to evaporate the solvents from our reactions, and then you get the desired product. So here this part is the condenser, and here this is the water bath, and this is the part where you attach your round bottom flask RBF over here just to evaporate the solvent. Then this is the UV chamber, which is used to view the TLCs, like the UV TLCs. You might have heard about thin layer chromatography. So this is like an advanced form of thin layer chromatography. We use the UV TLC plates. So this is a chamber in which UV TLCs are being observed under UV light. And moving further, that is the incubator in which we keep our things to get dry on. And then these are refrigerators and fridges to store our samples for the sample storage. So now moving on to the other part of the lab. So here, this is my colleague Andrew. He's working with the bone therapy. He'll tell you something about his work project.
Hello everyone, I'm one of Reddy's colleagues and I'm a third-year PhD student here at Queen's University Belfast. My work focuses on incorporating strontium metal into algae for bone regenerative medicine, but I also make synthetic counterparts, gels, as that's my background from my master's. As a gel chemist, I'll explain it in a little second. So we use sugar molecules, as seen here, to make a template in which we condense silica in a process called transcription. We then wash out the sugar molecule to leave the silicon nanotubules, and we hope to incorporate strontium in the same structure like this. The types of skills required to carry out this type of research would include NMR spectroscopy, IR spectroscopy, gas chromatography, atomic force microscopy, and basic lab skills. And that's it. Thank you.
Thank you so much, Andrew, for your speech. So now this is the freeze dryer machine. Basically, this freeze dryer is used to dry the samples under low temperature, like minus 20 degrees Celsius to minus 50 degrees Celsius. So these are the parts. We hang our RBFs or we keep our sample tubes inside this machine just to freeze dry the samples. So this is freeze dry. Hi folks, so now we are moving on to the photocatalysis lab. This is a photocatalysis lab in which light and catalyst are being used to break down the organic compounds. And here is my colleague Claire. She's working with the photocatalysis.
Hello, so my name is Claire and my PhD is about using photocatalysis to purify water and to break down contaminants in water. And so I started Queen's quite a few years ago. I did an integrated bachelor's and master's course in medicinal chemistry, but my master's project was kind of focused on organic chemistry instead. But then I moved on to this. So photocatalysis is a totally different subject. So it's really great being able to kind of switch between them and learn a lot of different interdisciplinary skills and kind of apply what I have learned into totally different topics. So yeah, this is kind of the typical setup that I use for the analysis of my experiments. So in here, we just have some water with trace amounts of different compounds like pesticides and antibiotics, things that end up in our water that we don't want in it, that's going to make it a lot kind of more impure than what we want it to be. So into that water, I just put some of the catalyst that we use. So the catalyst here, I think you might be able to see it. It's a white powder and it's called titanium dioxide. So this is used in like everything white that we have. It's used in white dyes and in like white markings on the road and in toothpaste. So we use that as a catalyst because it's really so cheap and it's so easily available. So all we do with that is put that into the water. So we'll put it in there and we'd let it mix up, and then we're going to shine a light on it, because when you shine a light on the catalyst, it creates a lot of free radicals and reactive oxygen species, so they kind of break down anything bad that they come across that we don't want. So this is an example of the lights I use. They're LED lights along a strip, so you see. So all we do is put the beaker inside that. There you go. So as the light shines onto the water and the catalyst, and then leave it there for a few hours and it will break down all of the contaminants in the water. So that's kind of the general experimental setup that we use, and this is how I analyse the results. So I use a thing called electrochemistry to analyse the results. So all it is you apply a voltage to an electrode, and then whenever the kind of contaminant that you're looking at touches the electrode, it produces a current, and that current equals the concentration of it. So it's a really good way to like monitor what's going on. So these are the little electrodes that I use. They're much cheaper than normal electrodes, like smaller or kind of larger electrodes. They're much smaller than the normal ones. And they're very cheap to make because they're screen printed electrodes. So they're just printed onto this bit of paper by a machine. So it's in here, and it's got the cables attaching it to the computer. So all I would do normally is I have electrolyte flowing the whole way through here all the time, and I just take some of my sample, some of the water, and we put it in here and squirt it out. So some of the sample is stored in this loop then. It's a 20 microliter loop, so it's a really tiny amount of the sample. So the sample goes through here and it's stored in here, and then whenever I open the valve, the sample ends up going over the top of the electrode, and that will give us a peak. So there we go. So we'll open it, okay, and we'll close it. Okay, so the sample has now run over the top of the electrode in here, and the signal is relayed back to the computer. So each time we do that, we get a different peak appearing here. So the height of each peak equals the concentration. So you can see at the start, we're kind of starting up at the very high concentration, and then after a few minutes even, it starts to decrease an awful lot. So that's how I analyse my results, and this is electrochemistry. So it's totally different than anything I did in my undergraduate or my master's, but it's really interesting to be able to learn new skills and kind of apply what I have learned before to this. So yeah, I would definitely recommend. You don't need to stick to the kind of subject that you started at. We have a range of biologists and physicists and engineers and chemists all working in this lab together. So there definitely is potential if you like the idea of it, even if you haven't done chemistry as an undergraduate. So yeah, good luck.
Thank you so much, Claire, for such a lovely speech. So here, folks, okay. We are moving on to the main instrument room. Here we have HPLC and fluorimeter. This is HPLC machine.
Hi.
And this is Steven.
Hi. So this is the HPLC RID. So this measures my reactants and compounds of carbohydrates and formic acid, and I'm able to monitor the concentrations over the time, and it works by measuring the output from the column in here. So It's the column. And then this has acid. This is an acid column, so it separates the carbohydrates and the formic acid. And then it runs through the detector, which uses light to reflect light at different degrees, so then it can be able to detect the quantities of our products.
Thank you so much.
Hello.
So now we are moving on to the fluorimeter. This is Ryan, and he's working with a fluorimeter to detect the impurities in the industrial waste.
Hi, this is the spectrophotometer, and I'm using it in my research to detect hydrocarbons in water. And this is going to be used for an online measurement probe, and hopefully this will give the company that I'm working for a lot more insight into the behavior of these compounds in water.
Thank you so much. So now he's going to show you how to measure the fluorescence spectrum of the compounds. This is the cuvette, and
this is a spectrum of phenanthrene, a common contaminant in water from oil pollution, and this is measured in the range 340 to 500 nanometers. And you should get a spectrum usually takes about 5 minutes to run, but it all depends on your parameters that you set up in your experiment. So it should look something like this, where you have peaks, and you can pick each one and find the fluorescence intensity of each peak. Thank you.
now we are moving on to the NMR Lab. These are the two different NMR instruments, one with the frequency 400 megahertz and the other one with the 600 megahertz. And this is the computer on which we get the recorded NMR signals. So from the basic, these are the tubes on which the NMR tubes are being placed, and then we put the NMR tubes over here, as you can see. So the machine takes over these NMR tubes with the sample, and you might have read in the NMR about the principle of the excitation of electrons to the external magnetic field. So we get the protonic signal, and the spectrum is recorded here in the computer. So this is NMR with the 400 megahertz frequency, and this is the one with the 600 megahertz frequency. Just the difference is that the precision of the spectrum which we get from the 600 one is much more clear than the 400 one. So otherwise, the system working is exactly the same. You can see there is a rolling machine with NMR tubes here, and exactly the same rolling machine with the NMR tubes. It's for the 600. It's just covered in 600, but it's not covered here. That's the difference. Otherwise, it's the same. So this was the NMR Lab.
So this is the chemical engineering lab with the supercritical CO2 extraction system. So this is Reedy's rig. So Reedy's been working on this for quite a bit. So
I'm working with the supercritical carbon dioxide extraction. Basically, I'm extracting the bioactive components from the seaweed samples. Seaweeds basically are the marine algae components, from where we will extract the bioactive components using this rig. These are the several vessels of the supercritical rig. This is the vessel in which I store my sample, and the carbon dioxide is flown through this vessel, and then the extracts that I collect comes through this collector vessel, and there is a carbon dioxide cylinder connected to the other side of the machine. This is the carbon dioxide cylinder in which liquefied carbon dioxide is being stored, and then the compounds are extracted. After extraction, these compounds will be used as animal feed to reduce the methane emissions from the livestock species. Thank you. This is a chemical engineering lab, and here
Hi, my name is Ralph. I'm one of the research associates here. And my area of research is in sustainable energy and renewable hydrogen. And beside me here is one of our experimental rigs that's investigating high temperature fuel cell operation. So you can see just below me here is a solid oxide fuel cell. So that consists of a ceramic electrolyte, which we use to either input hydrogen and get energy and heat out, or we can reverse that process and put energy in and we get hydrogen out if we use water as an input for electrolysis. This furnace behind us has been modified so that we can actually run tests on the kinetics and the operation and things like the performance curve of the fuel cell, so we can compare it to engine systems. So things like diesel generators, car engines, that kind of stuff, because some of the work that we do is looking at how we can integrate systems for industry and for transport with hydrogen energy. So you can see here we have a full setup that looks at combustion and the kinetics of the reactor. We're also looking at the energy intake down here. It takes to reform things like natural gas and ammonia into hydrogen for use in the fuel cell. And some of that work has formed the basis of developing a simulation of a large-scale hydrogen network that we're applying to Northern Ireland so that we can start to inform businesses and regional governments to incentivise and access some of that information to start companies and industrial processes out of it.
Thank you. These are our chemistry labs. Now we are moving on to the XRD machine.
We'll show like several peaks will show up, and then we'll fit up with the database. And the core components we're using is the zero background disk, which is made of the 9N semiconductor silicon. The operation is very simple. It's divided into two steps. We prepare for the sample first, and then we test it in the XRD machine. So I will just show about how to use the XRD machine. So firstly, give some samples here. As soon as possible. Then we put it into the center area of the machine. Lift it up and close the door. Overall, this is the procedure of the PXRD using. Thank you for your listening.
Thank you so much. This was the functioning of XRD diffraction study, in which we measure the solid state substances. Thank you so much for your talk.
This was the virtual tour, which was covered by Ms. Riddhi Salutra. So over to Ms. Riddhi.
Hello. So I just have one slide left just to explain a bit about my work.
Yeah, sure.
Yeah, so you guys might have already listened to the video. I already told a bit about my work. So basically what I'm doing is there is a process called methanogenesis that occurs inside the rumen of the kettle or the sheep. And due to that process, lots of methane is produced. And it has been estimated that 44% of these methane emissions, they contribute towards the global warming. So what is the main purpose is that I am actually extracting the bioactive components from the seaweeds, which are marine algae. So these bioactive components, they are being extracted using the green extraction methodology that I stated in that video was supercritical carbon dioxide extraction. So that is a green technology because it doesn't use any organic solvent. It just uses liquefied carbon dioxide as a solvent to extract the good components from the seaweed species. And those components, they will be used as tablets in the form of supplements, and they will be provided to the different livestock species just to see that how much dosage of those tablets or the supplements can reduce the methane emissions from the rumen and can help in reducing the global warming. So this is just a brief outline about my project that I'm currently pursuing at Queen's University. And this project is a part of Sea Solution project, which is a European Union funded worldwide project. So our main focus is to reduce the methane emissions from the livestock species using seaweeds extracts. So thank you so much for listening to the talk. And if you have any questions, you can contact me through my email ID. Thank you. Over to Narendra ma'am.
Thank you so much, Ms. Riddhi. So it was indeed very enlightening for our students. So for vote of thanks, I would like to invite our head PG Department of Chemistry, Dr. Manju Sani, to give a formal vote of thanks.
Am I audible?
Yes.
Yes. Hi, dear Niridhi, at the outset, and I am very glad to meet you. I am very glad to meet you. And at the outset, I am very, very happy to know about your achievement at this age.
Thank you so
On
much,
behalf of the Department of Chemistry, we sincerely appreciate you for taking out time from your busy schedule to provide us with such valuable information. All thanks to your support. You explained everything in a very nice and very crisp manner, and believe me, it would go a long way in boosting up our students. We wish you good luck for your future projects and look forward to meet you again. Thanks once again.
Thank you so much, ma'am, for your kind note. Thank you so much. It's really appreciable. Thank you.
So thank you once again, Ms. Riddhi, for really enlightening us with such a vast funding opportunities we have for our students. So students will also be happy to, I mean, they will be very happy to listen to you, right? Looking forward to you for more such informations from your side.
Thank you so much, ma'am. Thank you so much.
Thank you so much. And if our students will have any questions, we will just email you and
yeah, sure, ma'am. And I will share some documents like I stated before in the slides, just for the reference for the students to prepare their certification.
Yes.
Okay.
Sure.
Thank you so much, ma'am. And
Thank
all the
you
very
so
best
much.
to all the students for your future.
Thank you.
Thank you, ma'am.
Going to end the meeting.